Sir, you can start now, sir.
Yes, ma'am. Thank you. Thank you so much.
Good evening, sir.
Good evening, Anna. Good evening, Ajay,
Good
Dhanalakshmi.
evening, sir.
Good evening, Varsa also. Your new welcome, isn't it? Okay. Ritika. Okay. So last class, what we have discussed, my dear students? Will you please? Anybody else? Do you know? Ajay, will you please summarize the last class portion or chapter or say headings? Do you know? Yeah. What about Dhanalakshmi? Okay, anyhow. Today, what we are going to discuss about the topic is known as Principles of Inheritance and Variation, Part 5. Okay. So, first, before that, we must know what is meant by polygenic inheritance. What is the meaning of polygenic? Poly means already you know about more
Sir,
than.
Yes, sir.
Can you hear my voice, my dear?
Yes, sir. Yes, sir.
Okay. Polygenic inheritance. Already you know about inheritance. Inheritance is nothing but the genes or chromosome. It has a specific characteristics, features. That characteristics also transfer from one generation to another generation. That is called as inheritance, isn't it? Polygenic means poly, more than one, like that. Genic means genes. Inheritance means more than one gene inherited together. Okay. That is also sometimes that may be controlled, sometimes it may be express itself. Okay, that condition is called as polygenic inheritance. Once again, see here, the inheritance of the characters in which one character is controlled by many genes, and the intensity of the character depends upon their number of dominant alleles. The polygenic means nothing but you know about there are so many genes also present in a single locus, isn't it? It can be controlled by a single gene. The many gene controlled by a single gene. The condition is called as what is it? Polygenic inheritance. Okay, the characters should be will be how it will be, whether it be dominant or recessive, always in the dominant alleles only, but the number of alleles also very, very important. Character is also, it depends upon the number of dominant alleles. How many numbers of dominant alleles present in the locus? 100, take an example of 100 alleles is present in it. 60% is dominant and 40% is recessive, you like imagine. So 60 number is the dominant one. It can be expressed in the next generation in the form of polygenic inheritance. Okay. So this was first introduced by the name of the scientist. Take notes, my dear students. Polygenic inheritance first described by Nelson Ehil in Journal Color of Wheat. Kernel, actually, this is the yeah.
Sir, this Nelson Ehel is one scientist or two different scientists, sir? So
Nelson Ehel is also one, I think, no? It's a full name. Okay.
Yes, sir.
Okay. In kernel, the J is not coming here. It will not come. Kernel color of wheat. Note it down, my dear student. Wheat is an example taken by the Nelson Ehel for the process of polygenic inheritance. He observed, okay, the polygenic inheritance first described. At the same time, it explained by the name of the scientist called as Nelson Ehel in Kernel color of wheat. Nelson Ehel said that a kernel color of the wheat is regulated by two pair of genes. Here the question is that NEET question. Nelson Ehel, okay, in kernel color, how many pair of gene is regulated? Like that they may ask the question. So two pair of genes, that is equal to 4 genes. Nothing but the inheritance of the characters in which one character is controlled by the many genes, and the intensity of the character depends upon the number of dominant alleles. This was first introduced by the name of the scientist called as Nelson Nagel in where? The kernel color of the wheat. How many gene is present in it? Total 4 gene is there, 2 pair of gene, okay, respectively. Don't forget, my dear students. Okay. So next we will pass on to the next one is sex determinations. This is very, very important. Most of the cases, most of the question paper, it will be. Okay. So sex determination. See here, the establishment of the sex through the differential development in an individual at an early stage of life is called as sex determinations. You know very well about the sex is determined. So many processes there at the time of fertilization, after the fertilization, before fertilization, like that it comes. So many things is there. Okay. See here, the various methods operate in sex determination like environmental, non-allosomic genetic determination, allosomic sex determination, and haploidy, diploidy, diploidy, like that. Haplodiploidy, like that. That means it started from the N and end with the 2N category. You know about that male as well as a female. They are having the chromosome 46, isn't it? When it produces the gamete 23, and also the male gamete is also 23 and female gamete is also 23. So that condition is called as haploid. Okay. When it fused together during the formation of zygote, what happened? You know, the syngamy takes place. Syngamy means fusion of male as well as the female gamete united together form as a diploid zygote. So started from haploid and end with the diploid. So that condition is called as haploid diploid. Finally, what happened? Undergoes the meiosis process. They will get diploid. What is that? Child or fetus. Okay, that is last one. Okay, go to the next one. See here, you must note it down. There are 2 types of chromosome is present in it. Okay, for example, male is expressed XX. Suppose both the characters, homozygous conditions, both the chromosomes are X, it is denoted as female. Suppose X and Y is there. So X already know very well. The X comes from where, female part. The Y is the determining factor of what is that? Male. Already know. So the X chromosome as well as the Y chromosome also discovered by a different scientist. Note it down, my dear student. It is also very, very important. Okay, who discovered X chromosome? Who discovered Y chromosome? Okay, how many genes also present in it? That things also sometimes they may ask the question. So here, Stevens 1902, note it down, my dear students. Stevens 1902 discovered the Y chromosome. The Y is the responsible for the male baby. Okay, and Hermann Henging discovered the X chromosome. So this is called as Stevens Y. Okay, the Hermann Henging discovered the X chromosome. Okay, so this is must. This is very, very important. The question point. Next one. The X chromosomes contain how many genes is there? 1100 genes also present in it. Among these, 231 genes only identified. Sometimes they may ask the questions. How many genes are present in it? 1100. How many genes are identified till now? 231 genes only. Remaining how many things is there? 800 something not yet discovered. Identify which is responsible, which characteristics. Okay, so that is very, very important. Next, the Wilson and Stevenson. Which one is there? No, the Wilson and Stevens. The Y chromosome. 1905 here has given, and here discovered 92. See what is that? 1905 put forward the chromosome theory. Chromosome theory was stated by the scientist called as Wilson and Stevens of the sex with the X and Y chromosome being named sex chromosomes. Okay. Chromosome theory of sex. Note it down. This is very, very important. Chromosome theory of sex. Already we studied chromosome theory of genetics also already we studied, isn't it? Watson and Burnett, isn't it? So here the chromosome theory of sex with the X and Y chromosome being named sex chromosome. Who? The name of the scientist called as Wilson and Stevens in the year 1905. Okay. Don't forget, my dear student. This is very, very important one. What are the next slide? Sex determinations on the basis of fertilization. According to the fertilization only, the determination is come to the conclusion, isn't it? Already told you before fertilization, after fertilization, that things also we'll discuss. Sometimes they may ask the question. Okay. See here, what are the things is that there are 3 types have been reported here. Many observation, after the many observation, they have reported how many things? 3. Okay, see, progamy, gamete, listen carefully. Okay, progamic. The sex determined before fertilization. Fusion of gamete before what happened, you know, the sex also determined whether it's a male, whether or a female. That condition is called as what is that? Progamic. Example has given drone in honeybee. Example, progamic, drone in honeybee. When it determined, sex determined before fertilization, the male gamete and female gamete fusion together, you know, before the fusion together, the sexes are determined. That condition is called as progamic. Okay. Syngamy already you know. Syngamy, what is the mean? Fusion at the time of fusion. That is very, very important. See that syngamic sex is determined during fertilization, during the time of fertilization, male as well as the female gamete. When it joined together, that time sex is also sometimes determined. That condition is called as syngamic. Mostly plant and animals also an examples. Most of the plant and animals, simply. And then third one is called as epigamic. You have to tell us PSE. Okay, BPSE. Epigamic. The sex is determined after fertilization. Example, female in honeybee. Here the male, drone in honeybee. Here the female in honeybee. So these are the examples, okay, has given, and also they are having corresponding name. Don't forget, my dear student. There are 3 types of fertilization have been reported. One is called as progamic, syngamic, and epigamic. Progamic means before. Syngamic means at the time. Epigamic means after fertilizations. Don't forget, my dear students. Any doubt in this area?
No, sir.
Okay. What about Varsa? Newcomer.
No doubt.
Okay. Just to follow it. Can you follow my words?
Yes, sir.
Okay. Just go to. Okay. Go to the next one. And chromosomes are of 2 types. According to the nature of chromosome, they may be divided into 2 types. One is called autosome. Another one is called as allosomes. What is by autosomes? Autosomes are also called as somatic chromosomes. These regulate somatic characteristics. What are the body characters, visual aspects, skin color, like that things. Okay. Allosomes are heterosomes or sex chromosomes. What is that? There are 2 types of chromosome present in your body. According to the nature, they may be divided into 2 types. One is called as autosome. Another one is called as allosome. Autosome is also called as somatic chromosomes. These regulate somatic characteristics, your skin color, hair color, eye color. Okay, that things. Outer physical effect. Allosome or heterosome or sex chromosome, already you know about. These chromosomes are associated with the sex determination. Term allosome and heterosomes were given by the Montgomery sex chromosome first discovered by McClellan in grasshopper. Who is the first discovered by this one? Who stated the statement? The sex determination term allosome and heterosomes. This word was given by, term was given by the name of scientist called as Montgomery. Sex chromosome first discovered by which one in a McClank in grasshopper. You know all about the grasshopper, isn't it? When you are studying the, what is that? Ecosystem. Grass eaten by grasshopper. Grasshopper eaten by frog. Frog eaten by, that is called as food chain. Okay. So just note it down here. You must note it down. 4 points here. 2 types of chromosome already know about: autosome, allosome. Autosome, otherwise called as somatic chromosomes. What they regulate? Somatic characteristics. Allosome, heterosome, sex chromosome. This term was first given by the name of the scientist called as, what is that? Montgomery. Sex chromosome first discovered by which animals? Like that they may ask the questions. McClank in the scientist in grasshopper is the specimens. Okay, for the next one. See the first one. XX, XY type, or just listen here. This is very, very important. The XX, XY type. Okay, there are so many types also given here. Here you must note in the XX type and the XY. So both are common, similar to that of the human being. XX female, but the XY is a male. But what here they have given? Just listen. Types also called as Ligenous type. Ligenous. Yep. Whatever it may be, the pronunciation will be differ. What is that? Lignous type. Okay, Lignous. This type sex determination first observed by Wilson Stevens in Lignous insect. That's why they have given as a Lignous type. Which insect? Lignous insect. That's why the name is also called as Lignous type. The Lignous types also divided into 2 types. Listen here. XX female. Listen here. XX chromosome present in India, that is denoted as a female. XY is a male type. First one. A. Okay. So in this type, the sex determinations, female is homogametic. See that homogamete means XX is a homogamete. And XY is a heterogamete, isn't it? That is given here. Okay. That is given. See, that is produce only one type of gametes. Which one? Female produce only one type of chromosome only. X only produced by the female, isn't it? Okay. What about here? X also produced as well as the Y also produced. So male is a heterogametic. Next point, that one is comes, isn't it? See here, the male is a heterogametic. The male produced 2 types of gametes. This is the one type. Okay. In male, X chromosome containing gamete is called as a gymnosperm, and Y chromosome containing gamete is called as angiosperm. Example: man and dioecious plant like a coccinia and also melandrium. Here you note it down, my dear students. Already told you, man it has a male gamete as well as a female gamete. Female, it consists of homozygous condition. X is there, and male is X and Y. So female produce only one type of gametes only. That is called as X gametes only. That is homogametic. And what about the male means? It may produce 2 types of gametes, X as well as the Y also. Okay. They have given as an example. X is a, what is that? The X chromosome contain gamete is called as gymnosperm. Y chromosome contain gamete is called as angiosperm. It is advanced conditions. What are the example has given? Man and dioecious plant, otherwise called as, what is that? Dioecious plant means male is also separate and female is also separate. Okay. Coccinia, melandrium. Note it down. This melandrium is very, very important. So many times they were asked the questions. Melandrium containing which type of chromosomes? Okay. XX or XY. Like that they may ask the questions. Okay. Don't forget. Coccinia also. See that one. This is the plant, melandrium. Okay. Melandrium plant. Just a white color flower. And also some of them are having some, what is that? Seeds also. Just like I think it's a dioecious. Sorry. It's also one of a plant only. Okay. See the leaf structures. Bifid nature. See that each and every corolla is also bifid nature, just like a symbol of what is that? Okay, heart. Go to the next one. Another type X. See here, XY is female. Before we studied which one? XX is female, but here XY is female, XX is male. or jet W is female and jet Z is male. Okay. So this is another type. The before previous one, what is that? See here, XX female, XY male. Okay. But here, what happened, XY is female, XX is male, inversely proportional. That is also reciprocal. That is called as reciprocal. Okay. What happened here? See here, in this type of sex determination, female is heterogametes. Already in our level, female is a heterogamete. Produce two types of gametes, and male individual is homogametic. That is produce one type of gamete. Okay. So this is also reversible of the previous one. Okay. It is found in some insects like butterflies, moth, and the vertebrates like bats, fishes, and also reptiles. This is also different one. In plant kingdom, this type of sex determinations is found in, note it down, my dear student. This is very, very important one. XY, female. Okay. So, Phrygaria and Eliter is nothing but the strawberry. The strawberry plant already in our is one, nothing but a small fruits. A red color fruit is also, it contains number of, what is that? Vitamins, particularly vitamin C is more high. Okay. See that this is the plant. Cherry. Phrygaria, Eliter, strawberries. Okay. This is the plant. Here, the female containing heterogametes. Male is a homogametes. Okay, got it, my dear students. Sometimes they may ask the questions. Botanical name of the strawberry. Okay, strawberry contain which type of, what is that? Sex determination, say XY or XX. XY is female, XX is a male. Reverse of the, which one human being. And then second one is XX is female, already we know about, and also XO is a male. Like, note it down, my dear students. First we studied XX and female, and second XY is a male, isn't it? First one. Second one is you have to shuffle this one. XY and X is a female. But in this category, XX is a female and XO is a male. This type is called as, what is that? Protiner type. Protiner type. Note it now, my dear students. Protiner type of chromosome which contain which one of the following, like that they may ask the question. XX, X0. XX is female, X0 is otherwise called XO is called as male. In this type of sex determination, deficiency of the one chromosome, because here the Y is absent in male. Note it down, my dear students. That's why X0 I told. Okay, don't forget. XX female and X0 is male. This type is called as proteinor type. Why it is called as proteinor means the male gametes chromosomes, one chromosome is completely absent. That is called as deficiency. So what happened here in this type of female is homogamete and the male is heterogamete. Here it will also produce X and 0, but that is a things. Okay. So don't forget, my destiny. XX female, X0 male. This type of phenotype, you will get it. That condition is called as, what is that? Proteinor type. Don't forget, my dear students. See that one. So this is, what is that? Jet W. Okay. This one is called as female. This one is hen, isn't it? Female type. 76 plus Jet W. What is the total? How much is there? 78 chromosome is there. Note it down, my dear student. Okay. What about here? Same is there, but you will get it Jet. So both are homozygous conditions. What about here? See that housefly contains 6 plus XX. So total 8 chromosome is there. What about here? XY. It's a male. This type, this part one, this side only male, this side only female. Okay. How many chromosome is there? Just imagine. Sometimes we see that X0. Suppose if it is honeybee, what happened? X0. The last one also 21 plus X. What about here? 21 plus another. So likewise, you have to calculate. Don't forget, my dear student. These are all comes under the headings known as, what is the proteinor type. Okay, don't forget. See that. Sex determination in human. Already you know well about sex determination, already we discussed. Okay, which gene is responsible for the sex determination? How it will be tested? That things also already we discussed. Already you know what is meant by amniotic fluid, my dear students. What about Ritika, Dhanalakshmi, Ajay, Varsa? What is my amniotic fluid?
Sir, sir, it's a fluid, sir, and
Anyone, please.
sir,
Sir, it is the fluid in which the fetal is located, sir.
What is the use of that one?
sir, shock absorption, sir, and
Go
Sir,
good, good.
sir, it absorbs shock and protects the fetus from any physical shock, sir.
Okay, then main one.
So it is used for sex determination, like with the
Sir,
amniotic fluid, we can find the
amniocentesis
sex of the
process, sir.
baby before birth.
Good, super, correct.
Sir,
Yeah, both are correct.
sir, it is
Ah.
used for amniocentesis, sir, to detect
That's
any
that point.
karyotypic defects.
Yeah, good. That point I asked. Because amniocentesis is nothing but the sex determination, isn't it? How will you determine the sex? Before birth, you are collecting the sample from the mother and also analyze some components. What component already we discussed. Okay, good, very good. Vasa as well as Ajay also very good. Dhanalakshmi also good. Super, good going. Okay, so sex determination in human, just listen here. They occur a special gene on differential region of Y chromosome of human called SRY gene, sex determining region of Y chromosomes. What is that? SRY. Okay, sex determining region is called as SR. Y denoted as Y chromosome in a gene. Don't forget, SRY gene. SRY gene is nothing but sex determining region on Y chromosome. Don't forget, my dear. This gene forms a proteinaceous factors called TDF. Sometimes they may ask a question in explanatory aspects. What is by TDF? Testis determining factor. Testis determining factor. Don't forget, my dears. TDF. Okay. The TDF, what is the response for the development of the male reproductive organs? So presence and absence of Y chromosome determine sex. Suppose the Y chromosome is determined by which one, the TDF factors. TDF, how it will be? It determines the Y chromosome, whether male or female, like that we can come to the conclusions. When you are going to test TDF test, you will get the answer whether the baby is male or female, like that, just like as a amniocentesis. Okay, it's another type. What is the next one? See that father, already know about what will be the chromosome X and Y, isn't it? The JIT alleles is present at heterogamous conditions. What about mother? Homogamous. It split, it gives homogametes only. X only it gives from female. Whatever here, heterozygous condition. Correspondingly, we'll get it. What will happen? XX, daughter, XY, son, XX, daughter, XY. And this one is called as F1 generation. Note it down, my dear students. When your cross was cross with the F1 into F1 in digametic cross or monohybrid cross in the genetic, okay, you will get it. What is that? 4 different things, isn't it? This is the sex determination. XY, father is always getting a XY chromosome. This is heterozygous condition. This one is homozygous. Here, the mother only produces the X gametes only. What about here? Here, you will get it 2. That is called as heterozygous conditions, heterogametes. So, fuse it together, you will get it this type of, what is that? Combinations. Okay. So, go to the next one. This is also already, we saw it. So, this is the chromosome X and Y. So, X is already in our but present in the female also. So, the X chromosome, this one is called as Y chromosome. So, here the one gamete is also present in it. One chromosome is also X chromosome. Whatever here, the same is that present in XY. Here, Z. Note it down, my dear student. Z. XY is a male, XX is a female. But in this category, and hen and cock, Z type and ZW type. You just go to the book, you will get the ideas. Okay, got it? Let's go to the next one. Sex determination implant. Before that one, what we studied, sex determination in man or human beings. Before that, what are the sex determination types is also there. Okay. See here, the sex determination in plants. This was first introduced by which plant? Who is the scientist? See, H.E. Warmick discovered the sex determination in Melandrium album. Note it down, my dear student. First discovered sex determination was discovered in the plant. Which one? Melandrium album. Otherwise called as album. Okay. Who is the name of the scientist? H.E. Warmick. Plant Melandrium. Y chromosome is long as compared to the X chromosome. It is very, very important. In Melandrium plant, which chromosome is very long? Sometimes they may ask the question. Which chromosome is very short? Okay. See here, in plant, sex chromosomes are found only in the unisexual plant. This was first stated by the name of scientist, Professor R.P. Roy. Gave the importance of Y chromosome in plant. He discovered the sex determination in Coccinia indica, family Cucurbitaceae. Don't forget, my dear. Two scientists also reported in different plants. One is Melandrium album. Okay, H.K. Warmick. What happened, according to this scientist, he stated the statement, which one, Melandrium Y chromosome is long as compared to the Melandrium X chromosome. Another scientist, Professor R.P. Roy, gave the importance of Y chromosome in plant. He discovered the sex determination in Coccinia indica, that is family Cucurbitaceae. See that one? The Y chromosome contain, note it down, my dear students. Varsha, Dhanalakshmi, Ajay, and also Ritika. Ajay, don't forget, my dear student, this one. The Y chromosome, it contains 4 regions. Don't forget. How many chromosomes? Y chromosome contain 4 regions. X chromosome contain 2 regions. It's also so many times they were asked. The total XY, total how many chromosome is the region is the 6 region is there. Each one it has a specific property. Each one it indicates the specific characteristics respectively. Don't forget, my dear. Okay, see here. So this is the XY chromosome. See, the Y chromosome it contains 4 regions. See here, the segment 1. They have denoted as a segment like that. Segment means a portion of the chromosome part. Segment 1, segment 2, segment 3, segment 4. It contains Y chromosome. So the 4 region is here. What about the X chromosome? Only the segment 5 and segment 6 is also present in it. Which one? Melandrium album. Who discovered? Mr. R.P. Roy. Don't forget, my dear. Okay, here you must note it down. Y chromosome is 4 region. Y is a long one. X is a 2 one. Like that. Imagine. Okay, don't forget. So different functions of these regions. What are the different functions? For example, X is also having, Y is also having regions. Each region, it has a specific function, particularly, see here, the region 1, female suppressor region. Don't forget, my dear. The region in 1 is where it present? Y chromosome. So, it suppresses the female characteristics. So, the female suppressor region is called as first region. And second region is male promoter region. This one is first female suppressor. Second one is male promoter region. Third region is called as homologous region. Fourth region, differential region of X chromosomes. Don't forget, these are all present in the which chromosome? X chromosome. See here. First region, second region, third region, fourth region. Like that. The first one is what is that? Female suppressor, male promoter. And third one is called as what is that? Male promoter regions, homologous regions, differential region of X chromosome. The last one is. What about the, see that first region? Female suppressor region, what they will do? See here, this region suppress the development of the female reproductive structures. Okay. The male chromosome, it contains, what is that? 4 segment or 4 region. Male, they will not produce female reproductive structure. Okay. Because the presence of the first region only, first region of the chromosome. It suppresses the female characteristics produced in the male. Second one is second region, male promoter. Already you know very about. A region initiate or start the development of the anther. Otherwise called as male promoter region means it induced or indicate or produce or initiate. What is that? Male characteristics, particularly anther. Okay. And third region, male fertility region. Already I told you. This one is also another one, homologous fertility. This region induced the further development. Which one? Here the initiation. This one is called as further development of the anther. Okay. So 3 region is given here. Once again, first region, female suppressor region. What happened? Suppress the female reproductive part. Second region, male promoter region. So initiate the male development, anther particularly. Third region, male fertility regions. It's further development of the anther. Okay. And then fifth region, the homologous regions. Fourth. Okay, sorry. This region help in the disjunction and also pairing of X and Y chromosome during meiosis. Where there is a crossing over takes place, this region only, XY chromosome during meiosis. Fifth regions, just imagine different region of the X chromosomes. This region induce the development of the female gonads. That's the reason. Suppose both are XX present, one, it contain what is that? Female gonads production, X chromosomes. So when one or more than one Y chromosome present, then plant is male, and in female, the plant Y chromosome is absent. Suppose, what is that? Presence of one or more Y chromosome present in it, what will happen? It will denote it as a male. Suppose Y chromosome is absent, that condition is called as what is that? Female. Okay, see that one. This is very, very important. Okay, go to the next one. And single gene mapping in neurospora. Note it down, my dear student. Single gene mapping in neurospora. Do you know where will you study the neurospora? Ajay, neurospora. Already you studied these things. Where? Any ideas? What about Dhanalakshmi? No idea?
Sir,
Yeah.
sir, it's a fungus, sir.
Yeah, good. Very good. Any ideas? What about Varsa? Do you have any ideas?
No, sir.
Okay. Anyhow. The neurospora is a fungi. Already you know about. You know about one gene, one enzyme hypothesis.
Yes, sir.
That things. Who stated the statement? Piddle and Tatum. Don't forget, one gene, one enzyme hypothesis also done by Piddle and Tatum, where they will use, this neurospora is also fungi, mostly genetic engineering, gene technology. Okay, they were used in laboratory aspects. Don't forget, my dear. See, in neurospora, centromere behave as a gene for mapping gene pair. Centromere is also act as a what is a gene for mapping gene pair. In such a case, distance of gene from the centromere is calculated by the calculating the percentage of the crossover between the centromere and gene. How they will calculate? No, because the presence of centromere, and from the centromere, how many crossing over takes place within the chromosome, within the gene. Okay, that is done by which one, in laboratory aspect. That condition is called as what is that? That process is called as single gene mapping in neurospora. Okay, see, this is an example of neurospora. Single gene mapping in neurospora, otherwise called as bread mold. Centromere behave as a gene for mapping gene pair. Don't forget, my dear, this is the question. So many times they were asked the questions. Which one? Centromere act as a gene mapping process or gene pair processing. Which one is an example? No. Neurospora is an example. It is also considered it is fungi, mostly used for the genetic information, genetically used, genetic engineering. And also one enzyme, one gene hypothesis is also done by neurospora. Okay, laboratory aspects. See, then we will pass on to the next one is genetic disorders. What are the genetic disorders? You know, there are so many things is there. How it will be happen? Genetic disorder, genetic, gene. Disorder means, you know about whether it may be a mutated, sometimes it may be deletion, addition, substitution, insertion, inversion, whatever it may be, frame shift, so many things is there. Genetic disorders, okay? How will you identify our parent also, our grandparent also having genetical disorder? With the help of the one of the graphical or diagrammatical representation is called as pedigree analysis. Okay. Ancestor history. Suppose your father is also is there, and grandfather is there, grandfather is there, or grandfather or grandfather having some difficulties. Take an example, myopia, sugar, like that, you know, whether present or not, how will you identify it? With the help of the pedigree analysis. Then only the calculation will be very, you will not get it, isn't it? Suppose if it is, when you are putting the pedigree analysis, you will get the correctors. Take an example, the blood group also. Already we discussed it, isn't it? Ajay and Dhanalakshmi also, we discussed.
Yes, sir.
Yeah. Your father having some other blood group, alleles is there, IA. So a mother is also IA, IO. So what will be the progeny? Well, likewise, already we did, isn't it? Likewise, see here, the study of ancestral history of man, of the transmission of the genetic characteristic. You also look a grandfather or father. Grandfather characters also segregated from the one generation to another generation. From one generation to another generation to the next is pedigree analysis. Okay. So what are the things they may add? Dwarfism. Dwarfism is nothing but the depends on the physical characters. It is also short one. Albinism. Albinism already you know about. Color blindness, hemophilia, etc. I mean, there are so many genetic disorder diseases also identified. Okay. Or genetically transmitted characters. Suppose father has what you know, diabetes. The son is also affected by diabetes. What is the reason? You know, it is also one of the genetic disorder. Okay. Sometimes what happens? Suppose father has the diabetes, son it will not affect means, but your son sometimes may affect your the grandson is also affected. What will happen there? So you are the carrier. So it comes from where, you know, female. That means your mother or grandmother, they are having which, you know, diabetes conditions. Okay, what are the next ones? To study and analyze them, a pedigree of genetic fact, data, and following symbols are used. In pedigree analysis, they were used so many symbols. Each one it has a specific symbol, round and square and triangle. Okay, and also there is a connection between, like as a which one, you know, computer. Computer we were used so many symbols. Likewise, the pedigree analysis also they were used. What is that? For the genetical identification or data, so many symbols they were used. What are they? See here one by one. So okay. Before that, we must know what is meant by genetic disorder. What are the types of having? See here, genetic disorder is divided into two things: Mendelian disorder, otherwise called as gene-related disorders. Another one is called as chromosomal disorder. Listen here, my dear student. Gene disorder, chromosomal disorder. Chromosome, it contains genes. Don't forget. Okay. Where there is a location is called as locus. Particular gene is also affected. Sometimes this total chromosome is also affected. Okay. So they divide into gene-related disorder. Another one is called as chromosomal related disorder. Okay. First we see the Mendelian disorder or gene-related disorder. The gene-related disorder is also divided into two types: gene mutations in autosome. Autosome means body cells. Gene mutation is sex chromosome. These are the things. Again, so here the recessive and dominant is also present. The gene mutation is also autosome. It has two types according to the dominant or recessive characteristic. The recessive triads, note it down, my dear student. This is very, very important. Recessive triads: phenylketonuria, sickle anemia. These are all are contained recessive triads in gene mutation and autosome body cells. Don't forget. But dominant triads, see here, achondroplasia, polydactyl. These are all dominant triads. Already we discussed dominant triads. Some of them are coming, isn't it? Some of them are, it comes under the headings recessive triads. Here, the recessive triads, phenylketonuria, sickle anemia. Here, the dominant triads are achondroplasia, polydactyl. These are all comes under the headings known as gene mutation, autosomes, body cells. Sex determinants mean hemophilia or color blindness. Hemophilia and color blindness. Okay, this is the part. And another part, chromosomal disorder also divided into the same autosomal abnormalities, the sex chromosomal abnormalities. Autosomal means body. Here, the Down syndrome is also one of autosomal abnormalities. And sex determination, Turner syndrome, Klinefelter syndrome, it is comes under the heading sex chromosome. That is also whether it may be XX or Y, or addition of chromosome or deletion of chromosome. Likewise. Don't forget, once again, genetic disorder, two things there, gene-related disorder or chromosome related. Again, further gene-related divided to gene mutation, autosome or sex chromosome. This one depends upon their character, recessive or dominant. Likewise, they divided and also having specific examples, phenylketonuria and sickle cell anemia, recessive triads, autosome. Dominant triads are achondroplasia, polydactyl. Okay, polydactyl. This is also so many times they were asked the questions. And hemophilia and color blindness comes under the heading gene mutation, sex chromosomes. Likewise, chromosomal disorder also divided into two things, is that autosome and sex chromosome. Autosomal abnormalities, Down syndrome, and sex chromosome, Turner syndrome and Klinefelter syndrome, respectively. Okay, my dear students, listen. See that there are so many simple is that. See here, when we are getting a small square, it is called as male. Suppose we are putting simple round only, female. Opposites is, listen here, suppose if it is fulfilled by the dark, affected individual. Suppose if it is a male, when you are putting like this, this is what is that? Affected one. The female is also here. When it is affected like this, you completely fulfill the color. That is called as affected one. Okay, suppose you are putting the connection between the small line here. This is matting. Okay. See, this is the parental reason, parent and children. So these are all father or mother. Male is a father, square. Round is a female. Okay. This is called as matting. You will get the progeny, whether it may be a male or female. Okay. And dizygotic twins. Dizygotic twins. What is the meaning of dizygotic twins? See here, male and female. Sometimes each delivery, they may get, which one, two types of gametes, two types of progeny, the male as well as female sometimes get. Monozygotic, this is denoted as two male only. See here, two male. Square is a male, isn't it? Defined. Sometimes you may put a small, suppose instead of some male, you can put female also, round. Monozygotic twins. Okay. Here see that, suppose when you are putting the number within it, 4, that means 4 male child, 3 female child. Okay, likewise. So already we discussed, affected only individual. Affected means when you are darkened inside, that is affected. So heterozygote for autosomal recessive. When you are putting the off-filled one, it is considered as a heterozygotic nature for autosomal recessive. Carrier of sex-linked recessive. This is called as when you are putting the dark small circle inside it. That is called as carrier of sex-linked recessive. Decased. This is otherwise called as decased. Okay. Spontaneous aberrations. This one is called as spontaneous aberration. When you are putting the triangle, it is spontaneous aberrations. Consensuous marriage. Consensuous marriage. You put as what is that? Here the only one symbol is here, isn't it? Matting. Here there is a equal type. So there are two line is also connected with the male as well as the female. That is called as consanguineous marriage. Sex unknown. Suppose you are putting the symbol like this, this is also sex is unknown. Suppose we are putting normal means it's a male. Suppose when you are putting the straight person, this considered as a sex is unknown one. Okay. Not only that, so many things is also that. See that male, two male, prepan male, and decased male, affected by history, examined male, and see that Fratimal twins, identical twins. See, the fratimal twins is also another one here. Identical twins means also same. It's also connected. So in between these one is called connected, isn't it? So there is a small link between these two. Here there is no connection. So suppose if it is like this, there is called as fratimal twins. Suppose the connection in between is called as identical twins. Adopted male. See, the symbol is A. Suppose when you are putting A inside it, and also there is a covering, just like a what is a hand. Okay, this is called adopted male. Female, same thing is the two female, propand female, decased female, affected by history female, examined female. See here, the plus is the here, minus is the. This is also a carrier. When you are putting the symbol inside, it's a darker one. There is a carrier. And fraternal twins and identical twins. The last one is called as adopted female. Sex is unknown. Lived one day. Otherwise called as lived one day. One day lived one. Stillbirth. Stillbirth. Miscarriage. Pregnancy. Put in the symbol like this. There is a discontinued layer. Discontinued dot. And also inside there is a P. Marriage. Already you know what about. Contagious marriage. This one. Extramarital matting. Yes. Divorced or separation. Suppose when you are putting the two line in between these two. Okay. Here, no children. These are the symbol they were used. Okay, my dear students. We will finish it up to these levels. I ask some questions. Okay. Just imagine. So I think, okay. The question part, which one of the following inheritance takes place in only male, my dear? Ajay, Dhanalakshmi, Varsa, already we discussed. Which one of the following inheritance takes place only by male? Varsa, do you know any idea? Nuclear, cytoplasm, codominant, and holoandric dominant. Holoandric, male, So the holoandric inheritance is the correct one. Okay. Nuclear, it's not possible. Cytoplasm, it's not possible. Codominant, both the character. Yeah.
Sir, it's the D, right, sir? Holoandric.
Yeah, it's the correct one. Holoandric inheritance is the correct one. Because only they ask the male, nuclear, both it will be present. Cytoplasm is also so. Codominance, both the dominant character will be expressed in the. Here they ask which one, male only. So the correct answer is holoandric inheritance. Go to the next one. Who discovered Y X chromosomes? Already we discussed. Now only Y and X chromosome. Which one is the correct one here?
Say A and B.
A and B.
Yeah, correct. A and B is the correct one.
So,
Y chromosome. Yeah. Y
so
chromosome.
it's Stevens and Herman Henking, right, sir?
Yeah. So the answer is Ajay. Answer
Option
is
B, sir. Option
B
B.
one. Yeah, good. So Y chromosome who discovered?
So Stevens.
Stevens.
So yeah.
X is Henking, sir.
Okay. So don't forget, my dear students. Y chromosome is Stevens. X chromosome is Herman Henking. Any doubt in this category? B denotes Y. Isn't it? Herman Henking, X. H. Correct or not? Okay. Go to the next one. Which of the following genotype represent in intersex Drosophila? Do you know the ideas? This part. Okay. So remaining things we will see the next classes. Which one of the following is a correct statement in sickle cell anemia, my dear students?
Sir.
Glutamic acid by
Sir,
valine.
option A, sir.
Option A.
Glutamic acid. Option A, sir. Glutamic acid by valine at the 6th position of beta globin chain of hemoglobin molecule.
Very good. Super. Correct only. Exactly. Good. So because the glutamic acid in the globulin molecule, hemoglobin is also already known about. There is a change is that. The changes indicate the sickle shape of the RBC, isn't it? How it will be changed, due to the internal changes in the protein molecule. Particularly, there are two protein molecules there. 2-alpha, 2 beta globulin is there. Particularly beta globulin. So many amino acids also there. The amino acids, particularly the 6th position of the beta globulin, the glutamic acid replaced by valine. Okay, then only what happened? No, the normal RBC also modified into sickle cell shape. Okay, this is the question. This is the question. Don't forget, my dear here. Glutamic acid replaced by valine. Don't forget. Second thing is, which position? 6th positions. Which molecule? Beta globulin, not alpha globulin. Where? Hemoglobin. Where it present? RBC. So the shape of the RBC will be sickle cell shape. So this condition is called as what is that? Sickle cell anemia. Last one. The single gene mapping is observed in the fungi of. Now only we studied. Which one?
Neurospora.
Spora.
Very good, Varsa. Very good, Dhanalakshmi. Ajay also good, super. So today we will finish it up to this level only. We will see the remaining things on the next classes. Okay. Okay, take care. See here, this one. So just listen here. So this is the that position. Okay, see here. So here 1, 2, 3, 4, 5, and 6. The 6th position, which one is present in glutamic acid, isn't it? Here, what happened now? There is a change. Okay, glutamic acid is replaced by valine. Which position? 6th position. This is the that's why they have put in the arrow, isn't it? Which position? 6th position. Hemoglobin also very important. Beta globulin. Don't forget, my dear students. Okay. So I will summarize all those things. We will do the next one. Okay. So today what we discussed. Will you please? What is meant by sex determinants? Before that, we studied the polygenic inheritance. Who stated the statement? Nelson A. Hill. Where and kernel of color of the wheat. Okay. And then second one is sex determinants already in our report. What is by sex determination? Already studied. Allosomic, autosomic is also, and also. Who discovered X chromosome? Who discovered Y chromosome? Y, just imagine. In between there is a V is there. V, Y. Okay. H. So H is a female. Okay. Both H is present here. XX is a female. Like that you imagine. You can calculate it. How many genes? 1100 genes. How many genes are identified? 231. 1, 2, 3 like that. Don't forget. It's also sometimes confused. Okay. 132. Okay. 231 or whatever it may be. 231 simply. Okay. So Wilson and Stevenson. Y chromosome being named as a sex chromosome. Okay. X and Y. And what are the types? Progamic, syngamic, epigamic. Okay. So that things. And what are the types of chromosome? Autosome, allosomes. Okay. Autosome, somatic chromosome, and also regulated somatic characteristics. Heterosome, sex chromosome, allosomes. What is that meaning? Sex determination. Who discovered first? Meg Lang in grasshoppers. Before that, what is that? The term given by allosome and heterosome. The term allosome, heterosome were given by Montgomery. Okay. So then XX type, XY type. That is called as Lignus type. XX female, XY male. Okay. Other plant also, melandrium. Don't forget. And also this is a plant, melandrium. Okay. And then XY female, XX male. Okay. W female and ZZ male. So here the homozygous male, here heterozygous female. Previous one, homozygous, what is that? Female. Okay. Don't forget. Which plant is an example of this type? A strawberry. So Fragaria eleator. Okay. That things also. And this is the examples. So X female and XY is also another type of Crotina type. Don't forget, my dear students. Here male gamete is a heterogamete and female is a homogamete. So this is the example. Already we saw it. How many chromosome is present? And don't forget, housefly 6 plus 2, 8. Here cock and hen 78. What about this one? Grasshopper 23 or 24? 24. So here 23 is there. So sex determination human already go. Which one is the example? Stry gene. A sex determination region on your Y chromosome. Don't forget Y chromosome. Which test? Test determining factors. Okay. Okay. That's all about, my dear students. We will finish it up to this level today. We will see the remaining things on the next class. Thank you. Good night. Bye.
Thank you, sir.
Thank you, Ganam. Thank you, ma'am.